then let's start. So I'm just so we're gonna focus on this bidentate ligand. That's C2O4 minus. It's going to be exactly the same. So I'll just copy paste the previous one. I'm going to remove the previous ligand. Let's just add one C2O4 minus one. So C2O4 minus one was it had 2 carbons. So there were 2 carbons present, and there was an oxygen with a charge of -1. There was another oxygen with a charge of -1, and they had these lone pairs. And you had double bond, and you had an oxygen here and an oxygen over here. So that's C2O4. C2O4. This one. It's got 2 lone pairs. This side, the 2 lone pairs, and they're going to get attracted to this copper +2. So the other ones can also get displaced by C2O4 minus. There would be another bidentate ligand that could possibly take its place. So same thing, O-1, they're connected together by 2 carbons. So carbon 1, carbon 2, double bond oxygen, and there is this one has -1 charge with its 2 lone pairs. This one also has -1 charge, and it also has its 2 lone pairs. So that's C2O4 -2. Now this thing is, I mean, you just need to have a general idea of what a bidentate ligand is. So this entire thing will have a charge of, at the moment, it's going to have a charge of -2. Why? Because the -1, copper is +2, and this molecule is neutral. So if you add up the charges, Bhaskar, that could also happen. I mean, there's no reason why that should not happen. But if you think of it this way, that copper is +2, this oxygen has lone pairs, but this one is more negatively charged. I mean, it's got more negative charge on it as well. So since it's got more negative charge, that is the one that's going to be more attracted to this copper +2. Okay, the actual reason is kind of more complex. We'll study that in organic actually, but hopefully you got the idea. Okay, this one is already negatively charged. So the negative charged oxygen is going to be more attracted to copper +2. Is that clear?
Yes, sir.
That's the idea of a bidentate ligand. You just need to have an idea of what it is. You'll be asked to draw. They'll give you some random molecule, and they'll just ask you to show what a bidentate ligand looks like. I'll move towards, because in the syllabus they did talk about polydentate ligand, which is they had talked about EDTA 4-1 ETA. So polydentate ligands are they are ligands that can form many dative bonds. Now, polydentate ligands are kind of complex molecules that can form many dative bonds, many coordinate dative bonds. With the metal cation. Now, a lot of organic molecules, especially biological molecules like your enzymes and most of your cells, etc., they've got, they have this thing like iron in your red blood cell. I'm going to talk about that later. So again, you can't really remember the structure of ETA. ETA is this thing. I'll just copy the structure, but I'll copy the whole diagram. ETA is a molecule. This one is a good example, so we'll just copy paste. 1 second. This is fine. Okay, let's just copy this one. Okay, now obviously you can't remember this, so they'll probably give that in your in your question, the whole thing is you just need to know the idea of what a polydentate ligand is. So the first thing is there are lots of lone pairs available at lots of different positions. It's a big molecule. There's a lone pair here. There's an oxygen -1 here. There's an N lone pair over here. So there are lots and lots of lone pairs available in this entire molecule. So it's a big molecule. So this bidentate molecule was kind of a small, tiny molecule compared to a polydentate ligand molecule. Now, what will happen is that if you have, I mean, the idea is that if you have a copper +2 ion or any metal cation, then this molecule and its lone pairs are going to get attracted to this copper +2 from all sides. So it's, I mean, it's going to be, it would be like, I mean, this eta molecule would kind of, would be like wrapping around the entire copper +2 and surrounding it from all sides. So that's the structure of EDTA, which has a charge of -2. Or simply you just call it EDTA. And I'm going to show you how the complex is going to be formed. So the idea of the complex is, I think this, that's something that's slightly simpler. Just a second. So this is kind of a better image. So you can see it's the same eta molecule. There's a metal cation that's stuck in the middle. The oxygen -1 bringing its lone pairs from this side. There's an oxygen -1 bringing its, so the molecule is really large and it's wrapping itself around the metal ion. The N lone pairs are getting attracted. There's another N lone pair getting attracted to it. Oxygen -1 coming from the bottom. So is the idea clear? So this is one ligand and it's completely surrounding the metal cation. The metal cation is kind of trapped in the middle of it. And different parts of the molecule are forming dative bonds from different sides. Is this clear? Okay, Bakyo, is this clear to everyone? Zaib, is this clear? Zuha?
Yes, sir.
So that's it. That's the general idea. I'll just extend this idea because I mean, no one is going to ask you. First thing, no one is going to ask you to draw this. Okay, I mean, they're going to ask you to draw a bidentate ligand. They're going to ask you to draw a monodentate ligand, but never this thing. I mean, it's kind of impossible to draw, even for me. Now the thing is, you see a lot of metal ions. For example, lots of biological molecules. The metal ions are usually trapped inside a molecule like this. I mean, you've always heard about iron in red blood cells in the heme group in hemoglobin. So iron ions are trapped in exactly the same way. So that's how metals exist inside the human body, and they have different functions and roles that they play. So for example, let's talk about the heme group. It's going to be, the heme group is kind of similar to this. It's very similar to this. If we can see, if you can get a good image, a three-dimensional image. So you can see a biological molecule is like, it's like a very complicated molecule. So you can see this one is kind of a, okay, this one is an even better version. Just a second. Okay, so again, just the general idea of what it's going to look like. Okay, why is the image so small? Let me. So here you can see a heme group. This is part of the structure of hemoglobin. So if you look at it carefully, you would notice that here in the middle, there's this, so it's kind of like a complex. The iron is stuck in the middle. So here's the Fe, which I think is +2. I mean, this is actually Fe +2. So this Fe +2 is trapped in the middle. There's N lone pairs that are getting attracted to this Fe. There's an N lone pair that's actually at the bottom as well. And how does it absorb oxygen? Because when oxygen molecules are floating around, the oxygen molecule has lone pairs. So what happens is that the lone pairs of oxygen, they get attracted to the, so they get attracted to the iron, which is stuck in the middle. One side is open. That's where the oxygen molecule gets absorbed. Carbon monoxide poisoning is if you get a carbon monoxide molecule, that dative bond is kind of really strong. So the available space kind of gets completely used up, and the bond becomes is a very strong bond that's formed. So carbon monoxide doesn't really leave it. Oxygen forms a weaker dative bond, so it's kind of easy to break. Is this clear?
Yes, sir.
So now we're not okay. So remember just the general idea of what a polydentate ligand is. You're going to meet them often in complex biological molecules, and they're present in those molecules. Now, the next thing is define the term complexes. We did that. Describe the geometry, bond angles, shapes of linear, square, planar, tetrahedral, octahedral complexes. So describe the geometry, shapes, and bond angles. We've already done that in AS. So linear is when you have just two dative bonds. That's linear. Square planar, you've got square planar. We're going to talk about those as well. Tetrahedral and octahedral shapes, we've already done that. I'll give you an example, just an example of the geometry of a linear and a square planar molecule. So we'll just quickly go over the shapes. So shapes of complexes, the ones that we haven't gone through are linear. So this one is linear when silver ions are mixed with ammonia, and there would be two of them. This particular complex is usually linear. So it's silver ions dissolved in NH3. So what happens is you've got a silver ion, NH3 has a lone pair. Those lone pairs are attracted to our silver ion, and they end up forming dative bonds. And it's a linear molecule. It's because there's just two dative bonds. So if you know the geometry and the shapes of and bond angles that you studied in AS, then the shape is considered to be, and this entire complex is going to have a +1 charge. Why? Because ammonia was neutral.
Sir,
yes.
why did only 2 molecules of ammonia got attracted to Ag? Ammonia is pretty small, so why not more of them?
It also depends on the size of the central ion as well. I mean, there are lots of factors actually. It also depends on the size of the middle atom as well. For example, if the middle atom is big and the ligands are really small, so the geometry could accommodate a lot more of those smaller ligands. Is that point clear?
Yes.
So it's a relative comparison. What if the middle one is like really tiny and the surrounding ones are kind of big? So you can't accommodate a lot of them, right? Is that clear?
Yes, sir.
So that's just one of the factors. I mean, it's the ratio of the sizes of the ions. So you can't really predict because the ratio of the sizes is always changing. So most of the time you just have to remember, I mean, by just looking at the molecule, you can't really always tell what type of complex or how many ligands could be formed. And there's another factor, and that factor has everything to do with this silver ion over here. I mean, silver iron is attracting lone pairs. So if it has so there's this factor that if this silver iron in the middle has a very strong attraction for lone pairs, it's going to attract more of them. So if the silver iron with the positive ion in the middle has a very low charge density, it doesn't really attract the lone pairs very strongly. So not a lot of ligands would surround it. Is that point also clear?
Yes, sir.
Okay, so there are a number of factors that determine that. So this one, just remember, is linear. We'll just do one example for this. Okay, we've already done octahedral and tetrahedral. We did the whole thing with copper and cobalt. So we don't have to repeat that. One of them was square planar. And let me just quickly check whether we have cisplatin or transplatin still. Just a second. That is so. One second. I think they've removed it. So cisplatin and transplatin. 1 second. Stereochemistry. It should have been cis isomer square planar. Yes, that's there. So square planar. So they were talking about a square planar shape. So we can talk about cisplatin specifically. So I'll give that example because that's still there. So I'll give you one example of a square planar molecule, and that is a platinum ion, which is surrounded by so now this is surrounded by this is known as cisplatin. And it's surrounded by Cl-1 and 2 NH3 molecules. So 2 NH3 molecules. Platinum has a charge of +2 in this case. Cl has a charge of -1, and the whole thing is neutral. So this is a square planar arrangement. It's all the angles are at 90 degrees. The overall charge is going to be 0. It's not going to have a charge. And this is known as a cisplatin molecule. It's platinum with 2 Cls and 2 NH3s. And you got to remember this one. The overall charge is 0. Now, and it's square planar. Now, one thing that you need to also understand the importance of this specific example that we did. And that specific importance is that, remember, cisplatin is a very popular anti-cancer. It's used in anti-cancer treatment. So it's an anti-cancer drug. And I'll briefly, very quickly describe how cisplatin works. For that, you would need some knowledge of biology, but it's going to be a very general knowledge of biology. So we'll just comment a bit on cisplatin. It's an anti-cancer drug. What it does is that cancer is when your cells become malignant, they rapidly multiply. The DNA gets damaged, and the cells stop functioning properly. So tumors develop and they start multiplying. So your body is constantly, your cells are constantly reproducing. They're constantly splitting up. New cells are being created. But every time a replica is created, it has to function in the same way. If you have a brain cell, it dies, or not, it doesn't die, it splits up, produces new brain cells. those newer brain cells, they should function in exactly the same way as the previous ones. But what happens over time is that all the information is coded in your DNA, and your DNA gets damaged. And over time, if your DNA gets damaged, the cells would stop behaving in a different way. And there would be harmful mutations that would happen within the DNA. Now, the DNA is it's like a ladder-shaped structure. Not going into a lot of detail, but your DNA is like a ladder-shaped structure, and it's got these nitrogen bases coming out of it. There are actually 4 nitrogen bases now, and this is an oversimplification. So it's a big molecule that's coming out of it. There are 4 types of nitrogen bases, GCAT. So it's a polymer with lots of nitrogen bases that are coming out of it. This is how an actual DNA molecule looks like. If I show you, so the DNA structure is kind of, this one is a good example. So this is how it actually looks like. It's a polymer chain. So you can think of this as the polymer chain. There's a chain that's going on. Don't care what's in it, but it's a chain. And you got all these nitrogen atoms. These are your bases that are coming out of it. Lots of nitrogen atoms, so which is why they're known as nitrogen bases, because N has lone pairs. So I'm just drawing an overly simplified molecule. You got these N lone pairs that are coming out of it. And it's a ladder-shaped structure. So there's another DNA complementary strand, and it's kind of the same. And every time between that strand, there are different nitrogen bases that are coming out of it. Now, all the information about protein synthesis is stored in the order in which these nitrogen bases exist. There would be thousands, billions of these nitrogen bases. There's going to be this very long ladder, and every nitrogen base kind of contains information in what order are proteins going to be synthesized in the human body. So anything that has to be made, any enzyme that has to be made, the human body looks for the information in the DNA structure and in this sequence. And this sequence should not get damaged. Now, when cell replication happens, what happens is that these DNA strands, they unzip. I mean, the strands that you see over here, they split up. I mean, this strand creates another cell. This strand over here creates another cell, and the cell eventually multiplies and splits up. Now, if the DNA is damaged, you don't want it to split up. You don't want it to replicate. You don't want this particular strand to go away, and you don't want it to create this other new cell. You don't want to do that, because that cell is damaged and that information has already been corrupted. So you don't want that cell to multiply, because it's going to produce a lot of useless cells which are not going to function properly. Okay? So is this idea clear, the general idea? Basa, is this clear? Nadine, Zoha? Zaib, is this clear?
Sir.
So what you don't want is, you don't want the cell to actually replicate. So you don't want it to, over time, you don't want this, then let me just DNA replication. That would be, I'll just copy a few things. So this is how, this is just a general idea of how a different cell is created, or the cells they split up. So the ladder-shaped structure, the strand splits up, a new cell is created, a new DNA strand is built up using the old one, the 2 pieces of the old ones. So you don't want them to actually unzip. So what cisplatin does is that this cisplatin molecule, the one that you see over here, it sees the lone pairs on N in those bases, and it goes and it binds to them. So it already had Cl-1, its lone pairs. There was another Cl-1 and its lone pairs. And on two sides, you had NH3. And a kind of ligand exchange happens. The lone pairs of the nitrogen bases, they get attracted to the platinum. The lone pairs of the nitrogen bases, they get attracted to this platinum +2. And in that way, you kind of get the same sort of complex, except that instead of having two NH3s, you now have the nitrogen bases that are stuck with it. And the strand is not going to unzip. So this is what cisplatin does. It goes into the cell, goes into the DNA of the cell, and completely locks the strands together. So platinum ions, they bind with the nitrogen bases, preventing DNA replication. So that's how you kill cancer cells. You don't allow them to actually replicate. But there's this huge challenge, and that challenge is, how do you figure out which one is the cancer cell? That's the biggest challenge. So in early stage cancers, the cancer is very localized, so you can actually exactly tell where the cancer cells are. But in later stage cancers, you don't know where the cancer is because the cancer has spread out. You got these corrupted DNA strands all over the human body, and they're replicating. The cells are replicating. So it's very hard to actually figure out which ones are the good cells and which ones are the bad cells that should not replicate. So this is one big challenge that people face with drug delivery, especially with anti-cancer drug delivery. So now moving on, is the importance of cisplatin clear?
Yes, sir.
So now the next thing is we have to do I would go for colors. Just a second. Did we E0 values we're not going to discuss right now. We already did this. Okay, so the previous ones, coordination number, we did that. Our linear square did all of this. Yes. Forget E0 for now. I mean, redox, we leave that for electrochemistry. Now, colors. Let's start talking about colors. Now, the big question is, and the big question that they usually give in every exam, and that is, remember, A2 exams are kind of more predictable because they have, they usually ask 10 questions. It's a 100-mark exam. So most of the time, every topic, they have to give questions on every topic. Since they're giving questions on every topic most of the time, because it's a 100-mark exam, so there are lots of questions. So most of the questions are going to be very typical questions. They're going to be, so almost all patterns are going to be very similar. Unlike AS, in AS in paper 1 and 2, they had a lot of times you got a lot of inorganic, a lot of organic. Sometimes they won't give you moles. Sometimes they would give you moles. So in AS, in exams, the topics were kind of not that predictable. They would leave out some topics. They would add or give more questions on certain topics. But since it's a big paper, so you get questions on everything. And most of the time, it's the same questions that you get on everything. So it's this is one of those questions. And that is, why are transition metals colored? The metals are not colored. Why are transition metal compounds or complexes colored specifically? So that's the big question. Why are they colored? And remember, it's always the complex that's colored. I'll give you an example, like copper sulfate anhydrous is white in color. It's not colored. So it's actually white in color. So copper sulfate anhydrous is white, but as soon as you add water to it, it dissociates and forms copper 2 plus aqueous and SO4 minus 2 aqueous. Then this thing, copper with 6 water molecules, that's the thing that is actually, so that's the thing that's actually colored. That is the one that's blue in color. So most of the time it's the complex that's colored. It's not the compound that's colored, but the complex, when it binds with ligands, that's the thing that's going to be colored. So the question is, why are they colored? And the answer to that is, I'm going to just copy one of the complexes that we have drawn earlier. Let's take the water one. Where's the water one? So let's go with this one. Okay, let's discuss why copper with 6 water molecules, why do you get a color? So let's talk about this one. Now, the first thing is, in the middle, I'm going to add in the charges. It's just to show the charges clearly. You had a copper plus 2. Now remember this thing that copper plus 2 is going to attract lone pairs from all sides. It's going to attract lone pairs from all sides. And let's keep the water molecules away for a second. Okay, let's say at the moment, I mean, they are there, but they haven't yet joined it. So you had a copper plus 2 and it's got these 6 sides. The water molecules are going to get attracted to it. Now, copper ion has a completely empty outer shell. I mean, all metal ions, the outer shell electrons are lost. In the case of copper, the fourth shell electrons are lost. So that means, now that means that the outer shell is completely empty. And the electronic configuration of copper plus 2 is like this. It's if I write down the electronic configuration, it's 1s2, 2s2, 2p6, 3s2, 3p6. It was 3d10 and 4s1, but now it's 3d9. And the fourth shell is the one that's empty. So the outer shell, you got an outer shell that's empty. So that's what copper plus 2 looks like. So all these water molecules and the lone pairs, they're going to get attracted to this outer shell. While you have this inner shells, all these inner shells, they've got all these electrons. The 3p is full with 6 electrons. The 3s is full with 2 electrons. 2p is full. The 3d is partially filled. This is one of the reasons. This is one of the definitions of what a transition metal is. That the 3d is partially full. Now the 3d is the inner shell. Now listen to this carefully. That the 3d are the electrons that are inside the shell, and we did discuss what the 3d orbital looks like. In the previous classes, we did discuss that the 3D has 5 orbitals. So you got electrons that are lying between the x and z axis, y and z axis, the x and y axis. You got electrons that are lying on the x and the y axis. You got electrons that are lying on the z axis. So you got some electrons that exist between the axis. You got some electrons which exist on the axis. So you got 5 different d subshells, d orbitals. Probability is between the axes, and sometimes the probability lies exactly on the axis. So I'm going to draw these overlapping d subshells onto this copper. So that means you've got electrons that would be present between the axes. Okay, there would be there would be electrons that would be between the x and y axis, x and z axis, y and z axis. So all these spaces between the axes, they would kind of be full of electrons. Remember, the 3D is almost complete. It's got 9 electrons. And then you would have electrons that would be lying right on the axis. So the green ones are the electrons that would be lying on the axis. And now this is not a 3-dimensional image. I'm just showing that the 3D has many different lobes. There would be some electrons that would be on the axis. There would be some electrons that would be between the axes. Think of this in a 3-dimensional way. Think of it as like if I draw all the d subshells together, d orbitals together, you would have all these lobes coming surrounding the nucleus. Electrons between the y and z axis, between the z and x axis, between the x and y axis, on the x and y axis, on the z axis. So all these spaces would be full of electrons. And there would be 9 electrons. In the case of copper, there would be 9 electrons. And this is how the d subshell would kind of look like, this specific d subshell with 9 electrons. Is this clear so far?
Yes, sir.
Okay. And also remember that this d subshell has nothing, absolutely nothing to do with these ligands, because this is the inner shell. So this entire copper has a charge of +2. I mean, this entire copper has a charge of +2 because it lost 2 electrons. So the copper +2 is going to attract all these lone pairs from ligands from all sides. So all these lone pairs are going to be attracted to the copper. Think of the d subshell and all the other subshells as the inner shell of the atom. So all these water molecules are coming in and they're attracted to the copper +2. So this circular thing is your copper +2 ion. That's your d subshell with 9 electrons. Is this clear so far up till this point? Do you get this point? Like, I haven't explained why they have colors yet, but the first very basic thing, when you have a metal complex, when you have a metal complex, the inner shell has all these orbitals, and the inner shell also has a d subshell, which is partially filled up. The d subshell is going to have many, many electrons, like 9 electrons. Some of them would be between the axis, some of them would be lying on the axis. So we're running out of time, so we're going to discuss this in the next class now. Okay, so I just gave you an idea of what the complex would look like and where exactly is the d subshell. So I've just drawn this. We'll discuss and continue with this in the next class then. Okay, and let's continue tomorrow now. Okay.
Allah Hafiz.
Okay, everyone, take care.